Good afternoon, sir. Yes, sir. You are audible, sir.
Thank you, ma'am.
Yes,
A very good afternoon to one and all. I welcome you all for today's webinar, Automation in Bioprocessing. Myself, Dr. Shashidhar KN. I am the professor and head of the Department of Biochemistry at Sri Devaraj Medical College, which is a constituent college of Sri Devaraj Academy of Higher Education and Research. Our institution is a distinguished premier institution for medical education. Since its beginning in the year 1986, our institution has been started, and every inch of the work that we do in our institution is to the devotion, dedication, and the quality of the teachers and the staff. The vision of the management, which has led to this notable achievement of starting as a medical college, in unitary medical college with extending to the deemed to be status university. And it gives me great pleasure to acknowledge the dynamic leadership of our beloved late Sri R.L. Jalapa sir, the chairman, the founder chairman, and the founder president of our institution, and also the management for having giving us an opportunity to conduct the scientific sessions at our institution. I welcome all the dignitaries, my dear fellow colleagues, respected President of our Trust and Honorable Chancellor, Sri G.H. Nagaraja, respected Vice President, Sri Rajendra J, respected Secretary of our Trust, Sri Hanumantraju Sir, Honorable In charge Vice Chancellor and Registrar, Dr. D.V.L.N. Prasad Sir, Dean Faculty of Medicine and Principal, Dr. Prabhakar, respected Medical Superintendent, Dr. Krishnappa Sir, for today's webinar. It's my proud privilege to welcome our resource person, Dr. Barkha Singhal. Madam is the assistant professor in School of Biotechnology. It gives me immense pleasure in introducing ma'am. Ma'am is working at Gautam Buddha University, Greater Noida. Ma'am has done her PhD from IIT Roorkee and gained more than 15 years of teaching and research experience. Her research interest includes the bioprocessing of industrially important metabolites, such as the biosurfactant production and plastic degrading enzymes. Ma'am, hope your knowledge contribution to the audience today will be a feast for them to go ahead with the bioprocessing techniques and various other things. Madam has published various research papers in international and national reputed journals. She has also contributed several textbooks and chapters in highly reputed publishing house. Ma'am has written contemporary scientific articles in biotechnology area in various nationalized scientific magazines for scientific communication, and she has supervised PhD and MTech students. Madam's contribution to the science speaks volumes, and it takes more than a day to say more about ma'am. However, let us go ahead with the presentation. Once again, it is our privilege and reputation to have Dr. Barkha Singhal to be with us for today's program for the title Automation in Bioprocessing webinar. Myself, on behalf of my fluorosis team and my department faculty, the Fluorosis Research and Referral Lab, we have established the Fluorosis Research and Referral Lab, which is conferred with Center of Excellence by Sri Devaraj Academy of Higher Education and Research. We have intramural as well as extramural grants to conduct research. And ma'am, your inputs with respect to the Fluorosis Research and Referral Laboratory, collaborating with your thrust area, the bioprocessing, the various enzymes that you are working with shall help in reducing the fluorosis levels, fluoride levels in the water in this area, as well as the plant production of the vegetables, fruits, and other things. Kolar is the land of silk, milk, and gold. Now the tomatoes, Kolar is also popular with the tomatoes. With your contribution and joining hands with us, with our Center of Excellence, shall enable the agriculturists, the floriculturists, the horticulturists, and the population of the Kolar district to take it further, ma'am. So I wish to be associated with me and my team wish to be associated with you. Kindly extend your support in taking these things further with respect to the research. Thank you, and over to you, ma'am. Ma'am, please unmute, ma'am. Please unmute.
Yeah.
Yeah, you are audible.
Yeah. My voice is audible?
Ma'am, you are audible. You are audible.
Yeah,
Please go.
Thank you very much, sir, for a nice introduction. And it's my privilege to associate with you, and your department is working well. So whatever the problems related to the bioprocessing aspect, I will definitely try to sort it out, whatever my expertise is. Today's webinars is automation and bioprocessing. So it's my privilege to connect with these medical students today. So as I discussed with Dr. Soumya, that today's stakeholders are majority of the medical students. So I hope my talk will deliver some newer aspects in the bioprocessing. And I'm sharing my slides for my talk. Okay, if my slides are visible?
Yes, ma'am. Yes, ma'am.
Yeah. Okay, so today's talk has already Dr. Sir is talking about, that is the automation in bioprocessing. So myself, Dr. Barkha, and this is my webinar agenda today. So what is bioprocessing? I don't know that how medical fraternity is actually introduced with these type of terms. So I have started the presentation. I made my presentation from basic to advanced one. So everybody, so those who are not connected, they can understand well that what are the like developments in the bioprocessing from history to till date. So what is bioprocessing? Bioprocessing evolution, bioprocessing products, unit operations in bioprocessing, automation trends in bioprocessing, and what the future holds in bioprocessing, as well as that is okay. So as how many of you, so because majority of these stakeholders are students, so I want to float one question that how many of you know that what actually actual bioprocessing is? So can anybody give answer to me in this forum? So today's the session is like I want a more interactive. It is not like that I am talking and you are listening. So it is good that if you have any questions, or it's a more towards more interactive session I want. So anybody can give the answer that what is the bioprocessing, though it is written in that, but what's your understanding about the bioprocessing? Okay, so it is actually bioprocessing is as the term actually processing as well as that is the bio. So all the processes of the living cell which are actually helpful for the creation of any kind of commercial metabolite which is helpful to the society, which is contributing towards the any society like humans or animals or plants or our ecosystem. So that particular technology or that particular actually concept is originating from, that is the bioprocessing. So it is a like valuable technology for harnessing or exploring the different kind of products by any kind of living cell, whether it is like bacteria, whether it is your virus, or whether it is of fungus, any kind of animal cell or plant cell, or that is the or that is the any cellular component, like we are talking about, that is the enzymes, we are talking about the antibiotics. So either we can harness from the any cellular metabolite, or you can harness the metabolite from the entire organism, can be used for the bioprocessing purpose. So if we go for, that is the history, that is the concept is new. No, it is not the actually novel concept. So it is a very old concept that is actually started from actually before Christ 2500. So as I pasted two pictures for you. So our like that is the very ancestors. You can see that such kind of, that is the liquid, or you can say that is the alcohol is the most primitive product of actually bioprocess. So they have shown that at the very ancient time, the people used to get that is the drinking the alcohol for the recreational as well as the different purposes, as well as the bread making process, also known by, that is the early historians. So they know the actually fermentation process, means from what is fermentation. So fermentation is nothing but the conversion of organic molecule into that is the any kind of product which is actually helpful for human purpose. Okay, so what happens? So this is a kind of like, that is the historical background, and it is the Somras is actually mentioned in our epic also. So these are the so bioprocess actually evolution comes from actually our epics, and later on when we move forward towards historical backgrounds. So Hippocrates is actually known as, that is the father of modern medicine. So they also know that yes, mold fungus present in, that is they, or the mold which is present in the bread has actually healing capacity, or the wound healing capacity. But yes, the recognition of the antibiotic is actually discovered when it is a serendipitous discovered by, that is the Alexander Fleming. So the recognition was actually 2000 years later, but yes, Hippocrates know that such kind of concept is there. So that is also a kind of like, that is the bioprocessing, that yes, you can harvest certain products from, that is the living cell. Then moving forward, that is the alchemy, like that is the from the chemistry background, or you can say that is the origin of, that is the chemistry. So in the picture you can show, you can see that there are like certain kind of potions as well as the elixirs are formed. So by using the distillation as well as the fermentation process, they also know that yes, the magical kind of solution can be prepared by forming this kind of elixirs as well as by fermentation and distillation. So the technology doesn't know, but yes, that is the evidence of that particular concept has been found in, that is the alchemy also. Later on, the actual industrial revolution has been started by the Louis Pasteur discovery. So he's a famous scientist, and he is also known as, that is the father of fermentation. So he discovered that is the microorganism present in the that is the in the environment, and then pasteurization, fermentation. So lots of work actually has been done by the Louis Pasteur as well as the Robert Koch. So the real history, or the, like you can say, that is the glorious period actually, or the recognition period of the fermentation has been started after the Louis Pasteur discovery. Then if we see, that is the major breakthrough, though it is covered in, like that is the what is the milestones of the bioprocess development. So if you see the first bioprocess complete description, that is has been given by in the 2400 BC. Then yeast under the microscope, and that is the Antony von Leeuwenhoek. Then the industrial production of antibiotics is the major like it is a golden period for the industrial biotechnology, when the antibiotic was discovered during World War II. Then you will find that 1940 to there is a still the 30 to 40 years gap here, that is in 1979. So what happens? The antibiotic discovery was, the penicillin was actually discovered by Alexander Fleming. So it was considered as, that is the wonder drug. But what happens? The supply of this antibiotic is not as much as appropriate as demand was high. So they don't know that how they can overproduce this penicillin from the cell. So so the real contribution comes when the Watson and Crick actually discovered the DNA in 1951, and after 1962, when the restriction enzyme, actually mol bio development restriction enzyme, has been developed. So scientists actually know that how the DNA can be cleaved as well, ligate and transformed into suitable host. So real actually industrial revolution takes place after that is the discovery of restriction enzyme. So in the year after the 62 to 10 years, it will take scientists to to study all the different aspects of the restriction, like genetic engineering, and the first 1979 monoclonal antibody production by the hybridoma cell technology is there, and then the recombinant insulin is produced by Eli Lilly company, and then the later on there is that is tremendous development, vaccine development, therapeutic monoclonal antibody, then the genome sequencing projects has been initiated, and then the saga is still going on, and there is a relentless actually discovery of so it is just that is the glimpses of the first recombinant insulin, which is produced by through that is the industrial scale production. So bioprocessing is so as we see that is the different milestones. So it is not actually so the product come from the lab, but it has to go to that is the industry. So it is a kind of multidisciplinary field. So single biotechnologist or single biologist cannot produce any kind of commercial metabolite, let me say frankly. So it is a collaborative effort, or it is a kind of like cooperative involvement of various disciplines. So it's a kind of wide umbrella where the multiple disciplines are coming together to create one product, one important metabolite for the commercial purpose, or for that is the societal use. So next come that is the what are the different type of actually products. So as the living cell diversity is there, we have a like wonderful diversity in the living cell. So we have different range of products which are actually coming from here, that is the bioprocessing. So different types of organic acids, alcoholic beverages. There is a huge section of that is the alcohol is one of the the major contributing towards that is the economy. Then amino acids are there, vitamins and nutraceuticals are there, antibiotics, biopharmaceutical, enzymes. So they have shared the different, they are a good market share. So bio-based products are there, and generally there are two important processes, fermentation as well as the biotransformation, which is actually responsible for the industrial scale process. So it is a majority of the products are actually covered through fermentation in the industry scale. So this is like fine and specialty chemicals are there, as well as the novel materials are also produced from the bioprocessing products. Okay. Then this is a kind of actually different kinds of antibiotics which are actually semi-synthetic. So natural antibiotics are there also. Means there are natural compounds which can be acting as antibiotics, or generally in the bioprocessing arena, semi-synthetic antibiotics which are actually produced at a larger scale. So glycopeptides, lipopeptides, and then there are different class of antibiotics which are actually synthesized nowadays. So after the discovery of penicillin, there are lots of molecules which are described, and nowadays we are actually facing the antimicrobial actually resistance. So there is a big problem for overwhelming utilization of actually antibiotics nowadays. But yes, it is a separate and major of actually bioprocessing. We cannot deny, in fact, that, and we cannot think of our health without actually antibiotics. So nowadays what happened, there are novel kind of antibiotics, or the novel antimicrobial compounds have been searched for that. So next category of this bioprocessing products are actually antimicrobial peptides, and these are antimicrobial peptides which are actually extracted for different types of microbes, and which has again different like biomedical applications. So all drug deliveries, anti-cancer, anti-tumor, immunomodulatory, signaling, contraceptive, and mitogenic agents. So these are again derived from that is the different bioprocessing products. Then comes the what are the different type of biopharmaceuticals. So nowadays there is a term introducing like biosimilars, biologics are there. So there is a big category of the compounds or the molecules or bio-based molecules which are acting as in the biopharmaceuticals. So they are actually categorized as monoclonal antibodies, vaccines, gene therapies, cell-based therapies, growth factors, as well as the recombinant proteins. So these are actually type of biopharmaceuticals. Then we comes that is the enzymes. So enzymes, we have like tremendous applications. So different types of actually enzymes are there which are commercially important, which is used highly in that is the different type of industry. Every industry, like from textile or food processing, detergents, pulp and paper, wastewater treatment, personal care and cosmetics. So there are tremendous, it is a like good market share of enzymes contributing towards that is our economic framework. And these are certain key companies which I have to mention, that is these are actually key companies which are producing these enzymes at a larger scale. So these are all actually produced through fermentation as well as the bioprocessing. So these are also one of that is the major products from this area. Then then the actually from 2005 onwards, when the biosimilar development has been actually gained into momentum, so different kinds of actually monoclonal antibodies have been come into pipeline. And as I indicated earlier also, that any kind of living cell can be actually explored for the production of industrially important metabolites. So these are the different type of actually cells. CHO means that is the CHO cells, Chinese hamster ovary cells. So the various types of actually cell lines have been used to gain or to explore or to produce this kind of actually antibodies. So these are the major biopharma companies which are actually producing antibodies, and these are the different types of actually monoclonal antibodies which are produced through the bioprocessing or at the industrial scale. These are actually need to mention that different type of expression systems. So in the bacteria, if we are talking about the prokaryotes, Escherichia coli is the one of the prominent hosts. But besides that, like various other microorganisms are also used, but it is the most thoroughly investigated industrial workhorse. In fact, so it is actually used as a research as well as the industrial purpose. Same with that is the Saccharomyces cerevisiae in the eukaryote, then the insect cell, transgenic plants, transgenic animals, as well as the mammalian cell lines. So if we categorize the industrial product or the bioprocessing product, it is actually categorized into three types. One is that is actually high value, and then one is known as that is the medium value product, and then it is known as that is the low value product. So whenever we are actually high value, or you can say that is the biotherapeutic kind of compounds or the molecules are there, then we actually use the mammalian cell as well as the transgenic animals kind of expression system are there. So there are different expression system which can be, so depending upon the volume, depending upon the status of the bioproducts, we can use a different type of expression system for particular that is the production. So this is the different expression host. Then I need to mention that what actually real bioprocessing is that. So it doesn't always involve that yes, you can grow the microbes in one particular media. So why don't we use the waste material as a resource or as a substrate to grow on the microbes, and they can convert these waste materials into value added products. So here I put one slide, that is the waste cooking oil is converted into that is the omega-3 fatty acid through the fermentation process by that is the fungus. So this is one kind of, this is actually a process where the waste is actually converted into value added products. And nowadays what happens, waste management is a big problem. So waste wealth from waste is a like hashtag nowadays. So it is so bioprocessing is contributing immensely in this area too. So various value added product can be obtained. So I have come up with 2 to 3 slides example. So any other, that is the coffee, green coffee leaves, as well as the flour, as well as the that is the coffee processing waste, spent coffee grounds. So all these are actually used to produce the different type of, so polyhydroxyalkanoids are known as actually bioplastic. Apart from that, these are actually all value-added products which are actually produced through the different microbial fermentation and all. Then bioenergy is actually one good sector which we are actually seeing the potential, and lots of actually industry waste, agricultural waste, oil crops, biofuel crops have been tremendously used to generate, that is the different type of biofuels like biohydrogen, bioethanol, biobutanol, and they are actually classified based upon their crops, based upon their process methodology, that is the first generation, second generation, third generation, as well as the fourth generation. So fourth generation actually deals with the advanced synthetic biology approaches, where the advanced genetic engineering techniques have been used to improve the strain. And while first generation is particularly for the cornstarch, second generation is for, that is the all lignocellulosic. So second generation is the most popular one because India is the country where lots of actually agricultural waste are there. So if it is can be converted through the fermentation technology through biofuels or bioethanol, biohydrogen, then it will be great. So today, in fact, India's bioenergy policy has like B20 phenomena, where the 20% of bioethanol is blended with your 80% of gasoline. So later on in the 2050, they have like, that is the 50-50% blending they have planned for that. So it all actually happens, or it actually all contributes towards that is the, so unless and until we go for a large scale production of the bioethanol, we cannot meet out the actually demand for the society. So this is another arena of, that is the bioprocessing products. So in a nutshell, or is in a summarized form, so we can categorize the different kinds of products which I actually have talked about, and accordingly the industrial biotechnology is actually divided into different color codes, like brown biotechnology is bioremediation, where the environment, different types of microbes or the microbial products can be used to degrade the different contaminants from the environment. Then the blue or blue biotechnology or the marine biotechnology, where the different marine, or in the South India, it is very actually research, exhaustive research is going on for the exploration of the, that is the marine organism for different like drugs, pigments, lipids, amino acids, as well as the enzyme and antibiotics. So this is a marine or the blue biotechnology. Then green biotechnology based upon the, that is the plants, where like biofertilizer, biopesticides, and the different that is the value added products can be created by the genetically or the transgenic plants. Then the red biotechnology, which is used for production of all, we have biopharmaceuticals, antibiotics, or medical related compounds. And then the yellow biotechnology, it is actually food related, where the nutraceuticals, different kinds of food additives have been prepared through the living cell only, is where the living or the living cell components have been used. So definitely when the such kind of system is used, so bioprocessing comes directly into that arena. So this is the wholesome idea that yes, what are the different types of how actually bioprocessing contributing in this society or in the economy. So this is an idea for sharing this slide. Now come to the how actually bioprocessing. So we have talked about the like historical development, and what are the products which are actually seen. We don't go deep into the actually how we can actually enter into the bioprocessing world. So these are, that is the unit operations. Unit operations means how from one cell from scratch to actually industry, how from like, that is the from very basic, from plant to actually market. Plant means that is the industry to actually market, how we can go through. So these are actually known as unit operations. So what actually do when we go for, that is the bioprocessing. So first actually point of first, that is the unit operation, is that is the selection of cell, that yes, what you want to actually produce. If you are if you want to produce a very high or you can say that is the high value biopharmaceutical, so definitely your choice will be animal cell or animal cell lines. But if you are like looking for an antibiotic, if you are looking for an enzyme, so you can select, that is the microbe. So selection of the cell means that yes, which cell you want to carry forward for, that is the industry scale. Now selection again has certain bottlenecks or certain that is the phenomena or certain concepts. Like one concept is there, yes, you have cell banks there, and in the every country, like in our India, that is the MTCC is there, NCCS Pune is there, NCIM Pune is there. So there is a like sort of cell collection bank where you can purchase the cell and then culture the cell, make the product, and then go for that is the industrial scale production. And like this is the one way. Another way is that yes, no, you can isolate the cell from the environmental pool, and then you go for the screening part and then find out that yes, your metabolite is secreted, which kind of cell is suitable for that is the highest maximum production of metabolite. So the objective of actually the bioprocessing, so it is not actually charity work, it's a kind of that is the industrial framework. So it also depends, it is a major focus on that as the at maximum amount of product can be harvested from the cell with a less input of actually cost. So, and the entire optimization or the entire processing can be done in such a way that yes, we cannot compromise the quality of the product as well, and where we can cut the cost so the product can be economic. So sustainability of the product is also dependent upon that is the economic value. So if the product is very much high value, it is particularly not meant for that is the general society. Okay, so selection of cell plays very, because it is a major that is the successful criteria, because whatever the cell you are choosing, the such type of product can be obtained from a large scale. So it is a, like that is the one of the initial step, and it is a crucial step. So it is like kind of that is the seed selection. So whatever the seed you are selected, the plant may come out as per your that is the condition. So selection of cell play a very important role. Nowadays, what happens when we isolate the cell, there are two approaches where you can go for the traditional or the conventional cultural based method, where you can take this the soil sample or the water sample, any environmental sample, dilute it, and then make the certain specific media, and then find out that yes, culture is there, colony is there, find out that identify is there, and this is a like normal workflow which is routinely carried out in the laboratory, routine laboratory practices. But one novel way actually nowadays, what happened, that is the metagenomics, or you can say that is the like it is said that the 1 gram of soil contains 1 gram of soil has like enormous, like 10 to the power 9 types of actually cells are there. So in the culturable method, which is routinely done through the laboratory practices, it only satisfy only the 1% of the cell population. So whatever we seeing on that Petri plates, whatever we seen on that is the laboratory scale, it accounts only 1%. So 99% of the cells are actually hidden. So to find that is the unseen majority, the nowadays the technique that's actually known as metagenomics. So one way of isolating the cell of your desired interest is from that is the metagenomics. So selection of the cell has a very important aspect. So if you do the selection of the cell, so it will take years to like find out the yes cell. So this cell I am going to keep for the entire study. So it is not very much easy task to select the cell unless and until the background literature has been done. So it has been done very much accurately. Then after the selection of cell, you have to screen. So if you are going for a ready-made cell, like you are taking the cell from the culture bank, there is no need of screening, no doubt about it. You can directly go, you can grow, you can optimize, and you can directly go for that is the further processing, which I written here. But yes, but if you are isolating the cell from the area environmental pool, either through the metagenomics or either through that is the routine conventional method, we have to screen. Screen means that is you have to find from the large pool of the microbes or from the large pool of cell to that is the narrow down. Yes, this is my lead cell which can satisfy my most requirement for production of my my desired metabolite. So that screening is the nothing but it is a combination of various like biochemical process, immunological process, microbial processes. So different types of tools and techniques are there for screening, and screening is again divided into qualitative versus that is the quantitative. Then after the selection as well as the screening of the cell, then the storage. See, why it is actually written in storage of cell, it is it is not so much very much important aspect like the why it is written here that storage is also one kind of unit operation. Yes, because if we want to store, if because the cell has the capacity to mutate generation to generation, so storage of the cell is very much important. So it is not very much wise to whenever you want to produce any kind of metabolite in an industry, so again you go for the isolation and then screening. So it merely wastage of time, wastage of your cost, infrastructure as well as the resources. So unless and until you store well in a proper conditions, so there are the industry actually doesn't run in a cost effective or a economic way. So it is one of the crucial factors why that's why it is one of the unit operations. Then after that, when your cell is ready after isolation versus that is the screening, improvement means strain engineering. Basically, it is a improvement means like how you can improve your cell to attain that is the maximum productivity. So frankly speaking, in the natural cell cycle or in a wild type, if you know this particular term well, so it has no capacity to produce that is the over like your desired amount. Means nature has given that is the balance system or the homeostasis system. So if it is producing the over beyond their capacity, it will not be good for their survival. It will either in the form of any kind of disease, either it or it is a kind of that is the anomalous system for that cell. So strain improvement is the kind of that is the strategies or is the kind of actually procedures or molecular biology or any kind of like that is the biochemical procedures uh, to meet out the strain requirement or the our productivity requirement without altering the normal physiological process of the cell. We cannot kill the cell or we cannot kill the strain to harness the metabolite. We only improve, we only modify, and the genetic engineering is the one of the most common. And nowadays, you know, there are like tremendous development in the uh, strain improvement strategies from like site-directed mutagenesis to we have come to this stage that is E. coli is like if you have heard about that is the Syn 3.0, that is the synthetic E. coli cell. So where the E. coli, certain genes of the E. coli have been taken and the complete cell have been formed. So what they said that yes, the scientists said that yes, whatever the like required genes are there, we took out from the E. coli genome, artificially synthesized it, and then we made that is the complete cell from the small amount of genes which are present in the E. coli, and that is termed as that is the Syn 3.0. So we have come up with like the technology has been advanced in such a way. So strain improvement is a very important strategy, and we have to improve the strain, no doubt, whenever we are going for the bioprocessing, because industry is looking for the more product and cell has a limited capacity. So to meet out that capacity, we have to improve the strain. We must improve that is the strain. Then after the strain improvement part, that is the designing of the media as well as the optimization is done. See, there are two parts are there. Designing of media means what are the best component which are actually well suited for the growth of the cell, because there is a one very important parameter to uh, estimate that is the uh, what we can say that is cellular growth. And next is that is the how much amount of components which is required to grow the cell maximally. So sometimes what happens like that is the if we say yes ma'am E. coli is grown and that is the 3 grams per liter. So I said that is no. Can you add that is the 10 grams per liter? So sugar is the favorite that substrate for the cell. So can I add that is the 10 grams per liter? But no, we cannot do such kind of process key as if it is required that is the 3 grams. So by overdosing the glucose concentration in the media, the cell will grow more. No, excess is of everything is like dangerous or the injurious to cell also. So there are like lots of actually cellular phenomena occurs like substrate inhibition, product inhibition, so the cell will not feel safe. So unless and until the cell is not growing maximally, your product is not synthesized. So media designing as well as the optimization. So again, the optimization is nowadays, the techniques are so much advanced. There are various kinds of statistical tools or the computational biology tools which are actually helpful. So the work which took years to optimize the media components as well as the design, nowadays it is like 2 to 3 months practice to complete this kind of exercises. So this is again one of that is the unit operations. Next is the inoculum development. So after the isolation screening, then the strain improvement, media. So the next step is that is the inoculum development. See, in the laboratory scale, what we are actually saying that is the primary culture, secondary culture. So our faculty used to, like teacher used to say that yes, prepare the secondary culture and then add in the flask or add in the media. But inoculum is actually, it is a logarithmic phase of the cell or logarithmic phase of the any kind of the cell. So when it added into that is the production media, or when it is added into the tank, or when it is added into the flask, then the maximal productivity takes place. So if you remember from your previous knowledge that cell has to pass through various phases of the growth cycle. One is actually known as lag. Lag means that is your station. Lag means like there is no growth or there is a kind of adaptation phase. Then log is actually known as exponential phase. Then comes to the stationary phase where the metabolism is slow, but cell death is not there, but the metabolism is slow and the cell is trying to maintain. And after that, that is the decline phase or the death phase is there. So always inoculum should always be ready, or inoculum preparation is always, we are looking for like repeated culturing, only that is the exponential growing cells are there. So the maximum productivity can be achieved. So you are actually reducing the time. So industry doesn't mean, or industry doesn't want like that is the long process can be, because they have to invest in their infrastructure, they have to invest in their resources as well as their consumables. So they want that yes, maximum productivity can be achieved with a less span of time. So this is a kind of that exercise that inoculum development takes place. Then again, the next step of the unit operation is that is the production at shake flask level. See, any industry doesn't come directly from like that is the road. It always like lots of actually R&D work is lagging behind. That means it is always like that is the lots of R&D work is actually involved and doing permutation and combinations in the research. So shake flask means you are going the production first, you have want to check whatever you have done till date, that yes, it is working in your like that is the 100 ml conical flask, or you can say that is the 200 ml or 500 ml of conical flask. And if it is working well, then you can think of that yes, we can go up to the certain level. But if it does not working as per your desire, as per like that is the proper methodology, you can improve the process at the shake flask level. So that's why it is, or a production at a shake flask level, or you can say that one more prominent term is that is known as that is the seed fermentation, or that is the seed production. So either terminology is correct. Then comes when the shake flask level, or when your like lab level is optimized, then the designing of the bioreactor as well as the you can go for that is the production done. So bioreactor is nothing but the it's a kind of special kind of premises, or you can say that it's a kind of vessel where the any biological processes can be carried out under controlled conditions. So what happens, or like what is the basic difference between the shake flask as well as the bioreactor? Can the shake flask be said to be a bioreactor? So bioreaction takes place in the shake flask, but it cannot be considered as bioreactor. Why? Because in the shake flask, certain conditions we can control, but all biological parameters, all environmental conditions which affects the biological process, we cannot control at this level. So that's why this is a big difference between the laboratory versus industry. And in fact, it is a major bottleneck also. So we have very much talked about like there is a considerable gap between the industry versus academia. So this is the only reason that yes, at a lab level the things are working, but at a industry level the things are not working. So designing of the bioreactor as well as the production takes place. So whenever our lab scale is like ready to or it is working fine, then we can go for towards that is the bioreactor. Then after this particular part from the selection of cell to designing of the bioreactor and production, this particular term is of bioprocessing is known as upstream processing, means where exclusively the attention is focused on, yes, from the cell, how the maximum production of your desired metabolite, whether it is of antibiotic, whether it is of primary metabolite, secondary metabolite, anything you want to do with the biological product. So this particular upstream processing deals with that is the this different selection of cell to like the designing of the bioreactor as well as the production at reactor level. So this particular term is actually known as upstream processing. So what next? So when the like 10 liter, 20 liter, 50 liters of the bioreactor is there, you have a culture and then you want to separate out your desired product. So see, product have been actually ready, but what happened, that product is ready to use, or you can sell the product, or you can directly transfer the product to the customers? No. So the bioreactor is only meant for, yes, under controlled conditions, how cell can give the maximum amount of product. Now next task is also there. Next task is that, yes, from the large voluminous amount of your culture, you have to narrow down and you have to finalize that your purification as well as the recovery of your desired product. See, because there are lots of processes which are simultaneously going on, and lots of metabolites are released during the normal physiological process. So if you want to like purify one single product of your interest, then you have to like rule out the possibility of contaminating products from gradual, and that particular actually uh, stream of science is actually known as downstream processing. So upstream means that is from low level to higher level, you are production you are taking for focus on, and from the large amount of the broth, or you can say that is the culture or the fermented broth, you are finalizing the or narrowing down to concentrate the your desired product. That is actually known as downstream processing. So purification as well as the recovery, product polishing as well as the formulation means the polished product. Ultimately, if I want insulin or anybody want insulin, so they don't want ki behind kya chal raha hai. It just they want ki yes, if I need one particular vial for that, how that is the, so if I am able to deliver that particular vial of insulin, so that is actually known as product polishing as well as the formulation, that what kind of formulation is required. And then that is the one important sector, that is the waste management. Waste management is like whatever the biological waste, so how the industry is actually treating. So usually in the bioprocessing industry, it is advisable, it is like legal issues that the zero waste policy, zero waste discharge policy is there. So they are trying to give, yes, that is the solid waste, how they are treating, or how they are discarding, or how they are actually channelizing the solid liquid as well as the gaseous waste. So it is also one important part of actually bioprocessing. So just making the product as well as purifying the product is not bioprocessing. Waste processing is also one important consideration in the bio-based industry. So this is just like that is the glimpses about your, that is the unit operations, means how the actually bioprocessing can do from your lab to that is the industry level. So this is one aspect. So I already stated that it has bioprocessing has two components. So see, this is the upstream part is like that is the cell media different optimization, then this is the fermentation at a reactor level, and this is like that is the downstream. So as I already indicated in my last slide in detail, that upstream versus that is the downstream process, combinedly it is said to be that is the bioprocessing. Now if we look closely towards that is the upstream processing. So as I indicated earlier in my previous slide, that from selection of cell to your designing of bioreactor, so it requires considerable rigorous what I can say, that is the rigorous parameters need to actually consider simultaneously. You have to do the cell engineering in the medium. You have to find out, like, yes, what are the concentration, composition, conversion rate, any kind of anti-foaming concept, shear stress aspect. Then the operational aspects, like how the reactor can be operated. There are 3 modes of operation. I am coming in the next slide, like batch, fed batch, continuous cultivation. And in the, what are the different types of bioreactors are there? So is the uniform is the one bioreactor holds for all animal cell, all microbes, all plant cell, or based upon the cellular physiology, based upon your cell or product type, you have to design the bioreactor what is there. So again, the bioreactor has like type, mixing, shear stress, oxygen supply. So this is a one another kind of consideration. Then the process monitoring. See, lots of, when the process is going at from the shake flask level to industry scale. So only in the shake flask we just do nothing. We add the culture in the media. We kept in the incubator shaker, and the next morning or after the certain time and interval, we took out the flask, take the OD, and analyze our product. This is like, that is the routine conventional exercises. So we cannot think of like, yes, how much pH is fluctuated, how the dissolved whatever the oxygen concentration inside the shake flask is there. What about that is the nutrient when it is get exhausted? What are the gas streams? How much amount of, if the product is released, or it is like the byproducts are more released, or what are the side components are there? And then the analytics means like the type of actually products which are actually released during the processing. So these all are actually taken consider. So one by one consideration is not very like, that is the Herculean task. But if we are considering the all these factors in a one go, then it is a very difficult task. So at the industrial scale, the process is actually going to be actually difficult. So optimization, that is the upstream processing, in the productivity as well as the high data. So all these things actually lead to the, if we are optimizing these such 6 parameters simultaneously, the productivity of the product as well as the title of the product is actually high. So this is the objective behind, that is the upstream process. Now downstream processing has also, again, it is more costly as compared with the upstream processing, because from purify, that is to purify the the most desired or the purified product from the large pool or the large voluminous of the different kinds of product, it is not very much easy actually task. So downstream process, again, it depends upon, like, that is the location of your desired metabolites. And if you're looking for enzyme production, so first you have to find out that yes, it is secreted inside whatever the normal cellular physiology. It is secreted extracellularly, means cytosol, or it is like located inside, that is the kind periplasm, or any kind of, that is the intracellular location. So if it is located in the intracellular location, so we cannot say cell to no, you can release, that is the extracellular. You have to disrupt the cell by different means, and then separation, then concentration, purification, as well as the formulation. So different types of methods are actually involved when we go for this. So whatever you have understood about, that is the bioanalytical, like gel chromatography, gas chromatography, ultrafiltration, diafiltration, centrifugation, all comes actually different downstreaming actually aspects. So this is the unit operations, or we can say that is, yes, different considerations when we are actually going to the downstream processing. So if we like combine this conventional bioprocessing, then we see that yes, from the how the Petri plate, one Petri plate, we can come forward toward, that is the reactor production by reactor. Like this is a Petri plate where our desired cell is stored. We talked about, that is the plate or stock culture at -80°, means that is the glycerol stocks are there. Then you revive the culture, you grow the culture at a shake flask level, then optimize, then inoculum preparation is there, then the production scale. And the downstream processing, yes, centrifugation and filtration, different precipitation. So if you are like going for us, here in this slide, the particular product is your biopharmaceutical. So so you can see that is the series of actually the chromatographic applications, or you can say series of different steps are there to finalize actually this particular product. So It's a cumbersome process to. I think you can have an idea that yes, it's a cumbersome process uh, to obtain one particular product actually at that is the industrial scale, and it involves not only the biotechnologists, it involves a different type of engineering background people, different type of economic background people to like estimate the total cost of that is the particular process and to construct the such kind of high end sensors or high end equipment for that particular. So that's why it is said to be that is the multidisciplinary approach. So this is a basic actually conventional bioprocessing. So I would like to mention here that is the yes, because we are dealing with that is the industrial biotechnology or the commercial aspect. So when we are talking about that is the bioreactor, so one bottleneck I show here that is the bioreactor type as well as the different considerations in the bioreactor play a very important role, and there is a considerable gap. That's why the majority of the failures occurs. So things are working in the lab but not at a reactor level, not at an industry level. So I must show that yes, what is the reactor design, what are the different components of the reactor, what are the different modes that the reactor can function inside the industry. So this is a general layout of actually bioreactor. So it is a kind of made up of usually large reactors are made up of steel, but small at the laboratory scale reactors can be made of actually glass. So it has, so if you can see that it is a kind of like closed premises where this is actually sparger. It is known as actually sparger, where that is used to supply that is the sterilized air inside. So the aseptic conditions or the sterilized conditions are necessary throughout the industry. So if any kind of contamination can take place inside the process, your entire efforts are actually get futile, and lots of actually cost is waste. So everything which are actually done inside the reactor, it is actually in the sterilized condition for the aseptic conditions. So air supply for that is the sparger. If you see this particular shaft-like structure, this is actually known as impeller, where it is used for actually mixing. Then it is a long blade-like structure, which is known as that is the baffle. And there are different types of supply acid base, because I already mentioned that there is a considerable difference between the conical flask as well as your reactor. So reactor is when we are saying that yes, the reaction, biological reactions are taking place in the reactor, it means that yes, it is under controlled conditions. So all environmental parameters like pH, temperature, oxygen, pressure, whatever you can think. So all things are actually controlled, and how it is controlled. There are like certain measurement as well as the controlling devices are kept for every environmental parameters are there. So you can see that is it for the temperature control, cooling water supply is there, thermal jacket is there, then the different type of sensors to check the temperature, aeration, as well as the pH, pressure gauges to maintain that is the pressure. So acid base is also, if the pH fluctuation is there, if we like suppose we want to carry out the process at the 7 pH for, and I am saying that my process is going to run at that is the 18 hours. So 18 hours, if we go in a like simple shake flask, what happens after the certain timer interval when the metabolites get secreted, either the pH can be increased or decreased. So we don't have any kind of like measurement system, or we cannot have a like any kind of maintenance system ki no, at the 18-hour supply all the like pH of the pH of that particular solution is maintained at that is the 7 due to, for the particular 18 hours whenever the process is actually going on. So acid as well as the base, if the like pH is below, then the acid base is added, and if it is high, then the acid is added, or like that is the whatever the process value is there. So acid base supply is there for the maintenance. Then the process is actually done at industrial scale at a 3 mode: batch, fed batch, as well as the continuous. And there are 2 types of actually fermentation processes: submerged versus that is the solid state. So the so majority of the industrial processes are actually gone through that is the submerged. Submerged means that is it is immersed in that is the liquid media. So this is actually this particularly pink framework is actually designated as the media, and your like cells are actually immersed in that particular media, and then the process is actually carried out. So this particularly Batch means that is the once the system is closed, when the cells media are kept inside the reactor, there is no exchange of anything between. So once you have close the like system, then you whatever your end time is there, you can open and you can harvest the product. So this is a kind of like that is the batch. One batch means like there is no disturbance in between. In the fed batch, what happens, you have to add, because see, when the nutrients are there, or when the cells started to grow inside the media, definitely there is a starvation of nutrients are there. So at a interval, certain interval of time, when we actually optimize in the lab scale key, yes, what is their log period, and when the cell actually reaches there, the cell is going to in the stationary phase. So before reaching that is the stationary phase, you are adding certain amount of that is the substrate, or like glucose or any kind of media component, which is helpful to cell to not reach up to the stationary phase, because stationary phase if it is reaches in the stationary phase, then the product is not as much as formed. So you are adding certain amount of that is the nutrient solution in between. So that is actually known as fed batch, but you are not taking out anything from that is the reactor. So that is actually, that mode is actually known as fed batch, means batch is actually fed intermittently. Now, continuous system is, as the name indicate, whatever the feed, like suppose you have a 10 liter of that is the reactor is working on. And after the 12 hours of fermentation started, you find that yes, the cell is going to reach at the stationary phase. So in the 10 liter, you are adding like suppose 100 ml of your glucose solution as a feed to like to satisfy the nutrient availability. Now, the same 100 ml of your media volume should be, or culture volume should be actually extracted out. So that type of process is actually known as that is the continuous mode of fermentation. So in the industry scale, what happens if you are looking for a particularly for the primary metabolite, continuous mode of manufacturing is more preferable. If you are looking for a like secondary metabolites, where secondary metabolites are generally produced during stationary phase, so we can actually go for that is the batch process. And 99% of the processes and like that is your monoclonal antibodies or high therapeutics, that is actually through that is the fed batch process. So industry scale has a like three modes of fermentation. So depending upon the cell, depending upon your cellular requirement and depending upon your product type, you can select any batch as well as that is the continuous process. Now, for your information, I have like come up with certain slides that yes, these are like topographic representation of the reactor. This is actually reactor. You can look industry scale. This is the reactor, different types of actually reactors. So this is also one reactor. So like for the alcohol fermentation. So depending upon the product type and market volume, like what is the volume which is required by the society, or so different types of reactors have been actually designed. So these are all different types of reactors are there. So for a particular plant, so I stated one statement in my earlier slide that yes, whether one reactor is suffice to cultivate all plant cell, microbial cell, animal cell, no. There are like depending upon the cell physiology, we have to construct our reactor in a different way. So this is a kind of actually photobioreactor where if exclusively it is meant for the microalgae or like cyanobacteria, or that is the plant. So in this particular photobioreactors, you have to give the intermittent light and day cycle, or like that is the light for maintaining the light and dark cycle of that is the plant. So these are the photobioreactors. Nowadays, what happens? For the higher end therapeutics, because the contamination or to avoiding any kind of risk of contamination or any kind of like error, single use bioreactors are also there. So if you see compare with this such so these are not actually single use, these are actually batch to batch, means one production has been started, then it is completed, then again it is cleaned and again it is used. But nowadays the technology has been so much advanced, so there is a like if you can see so it is made up of biodegradable plastic. So it is a kind of single-use bioreactor. So there is a small, like 5 liter, 100 liter, or 200 liter of like biopharmaceuticals has been prepared. So volume is not very much high, but it is a single use, means that the single time the reactor is used and it is discarded. So if you are looking for a very high-end biopharmaceuticals, then the such kind of technologies are also available. Then this particular two features are when the multiple reactors are connecting with each other to like optimize the various parameters in a one go. So I think you have heard about that is the microarray technology, where the multiple genes are expressed, or multiple gene expression can be seen in a one slide or one chip. So that kind of concepts actually come through, that is the high throughput bioreactors, where the, if you can see closely, so this is a one complete bioreactor. This is again, so these are series of bioreactors are and the capacity is only, that is the 1 liter or 500 microliters. So this is a micro bio reactors, or you can say that is the high throughput reactor. So if we are looking for a, like if you're developing certain novel component, novel compound, and you don't know the cellular physiology, so the such kind of actually framework can be used, but it definitely the cost is very much high. It is the very good pharma companies like Biocon, Serum Institute of India, or the major like that is the Biopharma R&D hub, they can actually afford to have such kind of high throughput bioreactors. So this is the different types of bioreactors. And this particular bioreactor design is for, that is the tissue engineering applications, means animal cells. So if you see the different type of, if you are like looking for a 3D imprinting or certain cartilage based bioprocessing, where these such kind of, so so you can, you see like different constructions. It is a inner flask, then rolling bioreactor, then wave bioreactor. So these are all particularly used for, such kind of framework is used for, where we cannot actually add, that is the agitator. So to mix the cell, because animal cells are very much fragile, because they don't have actually cell wall. So for the animal cell culture, we have in the literature there are various design consideration of, that is the various bioreactors. So this is the different class of bioreactor which is used in the cell, different type of actually cells. So what we like, what is the take home message from these slides, that yes, depending upon the cell type and depending upon the product type, we can construct the design. So there is no universal design of actually bioreactor. It can be from small scale to a higher scale. But yes, it depends upon like what type of have been selected for or chosen for our industrial production. Now, whatever we have seen till time, if we see that yes, it's a complicated aspect, or it is a like cumbersome process to to visualize each and every angle of from cellular framework to production framework at a very minute level. So we have to consider, that is the scale aspect. We have to work upon, that is the system, means whatever the cellular engineering framework, and then combining all these features, we have to maximize the productivity of, that is the your bioproduct. So definitely it is not exclusive, like it is not exclusively done by the biologist or the biotechnologist. So engineering term actually has been inculcated. So as we know that living cells are quite complex, okay, and to reduce the complexity, engineering, what is the like, what is the basic idea of it? What is the basic advantage of the engineering? Like engineering, engineers always works in, that is the large complex, or the large complex things into, that is the reducing the complexity. The basic idea behind engineering is to done, that is the to solving the complex problem into, that is the in a simpler way. So that's why the different engineering principles have been actually involved. So I am not saying that in the traditional bioprocessing which we have discussed till date, it doesn't involve engineering. See, without involvement of the engineers, these reactors, what you show inside, So this is not a task of actually routine biotechnologists to like construct such kind of reactors. So it is like all multidisciplinary task. Here actually I want to emphasize that when the biology is actually amalgamated with actually engineering principle, the complexity of the objective is to implying the engineering is to reduce the complexity, to understand more process in a comprehensive way, so the maximum productivity can be actually availed. So that's why as I indicated earlier, because shape doesn't meet your requirement, so we have to shift towards that is the in a industrial level. So when the shake flask is actually converted into the when the production scale is converted from shake flask to industry level, then the terminal that is the scale up. So scale up means from small scale production you are actually going to that is the large scale, and large scale has actually no limit as per the requirement of the product and what is your capacity to produce the product. So if you have a like money to invest like that is the 1,000 billion liters of there to producing the capacity, you and the market demand is high, so you can go up to that level. But if your pocket is not aligned, if your infrastructure is not aligned, you can restrict the production. So that's why I'm saying that at the large scale there is a no capacity, there is no end for production scale. But there are certain actually scale up challenges are there, which, and I repeatedly told in my talk that yes, there is a considerable difference between from the laboratory to like academia to industry. So in the academic lab is working well, but at the when it reaches actually at the industry, it is actually not working well. So if we see this particular graph, that basic research and then the market penetration, there is a one zone is there which is actually known as the valley of death, and if this particular valley of death is recovered or valley of death is covered, then the commercialization can take place. So if we have to actually uh, cross the valley of death to commercialize any kind of actually product. Okay, so what are the scale up issues? Tank aspect ratio. Tank aspect ratio means the cell is actually habitualized to grow in like 50 ml, 100 ml, to 200 ml, 500 ml. So when you are when you are transferring the cell into like the billions of liters or like the gallons of liters, the behavior of the cell is actually quite different, which is not expected in that is the shade flask. Pressure is one of that is the important factor. Then the oxygen transfer rate. See here we are keeping that is the in the incubator shaker, so we don't measure the oxygen. But here I told you in the reactor design there is a sparger which is actually connected for a particular air supply, but simply providing this bubble form of oxygen is not sufficient unless and until you are, so there is a synergism between your impeller, means that is the agitator versus that is the sparger. So the purpose of this bioreactor is to maintain the uniform condition or the homogeneous conditions throughout the process. So whether the process is carrying out like that is the 1 day or 1 month, the purpose of the reactor is to maintain that is the homogeneous conditions. Okay, so actually the oxygen transfer rate plays very important role, and it is said to be like that is the yardstick of the fermenter performance or yardstick of the bioreactor performance. Why? If the oxygen transfer rate of the particular product or particular process is actually optimized well, then we can say that is the 95% of our commercial success is there. So majority of the failures actually is actually done with that is the oxygen transfer rate, where you think that yes, I am supplying the proper oxygen, agitator has a machinery to mix the oxygen in the reactor, but cell is not able to uptake. So if the cell is not able to uptake, so that particular oxygen, the transfer rate is not actually sufficient, because ultimately objective of your oxygen oxygen is to that the cell is actually able to take the oxygen properly inside the reactor. So this is the major bottleneck, and majority of the failures are actually occurring due to the less due to that is the imbalance between the shake flask level to that is the industry scale in terms of oxygen transfer rate. Then the feed rate is there. Then the heat transfer, because when the such kind of like motorized, or you can say that is the automated workflows are there, so lots of heat is generated during such kind of like large voluminous process. So whether this heat can be suitably transferred, because living cells cannot tolerate or cannot work in that is the high heat or as well as the low temperature. So all conditions should be maintained, as well as that is the mixing speed, shear sensitivity, as well as that is the foaming. So when the media is voluminous, there is a lots of problem when the oxygen is supplied, though foam is there, and foam actually reduce the oxygen. So if the foam layer is created inside the reactor, so it actually minimizes the oxygen. Actually, it interferes in the oxygen supply. So these are the major scale up issues when we are going for the industry scale to laboratory scale to industry scale. So, why actually we go for actually scale up? So, objective is to like increase the tighter as well as the productivity in terms of like infrastructure as well as the large amount of product. Then cost with cost effectiveness is always there, because industry is always looking for like that is the sustainable process which is cost effective. So if their input is more and output is less, so there is a loss. There is not actually sustainable. They cannot be actually sustained long for in the market. Scalable, as well as that is the they have to convince that is the investors. So if we are like based upon this discussion, what we conclude that, so like if we want to go for like there is a successful like successful biomanufacturing or successful production of your desired metabolite, then we have a optimized process control. It means everything should be like meticulously think on optimized process control, enhanced product quality, coping with that variability, increased throughput as well as the efficiency, cost effectiveness, scalability, as well as that is the sustainability. So these are the like which are things which are actually need of an hour. And as per the as you know, in the pandemic also, or the post-pandemic scenario, and in fact pre-pandemic scenario also, there is a lots of actually awareness related to the bio-based products are there. So everybody knows that the bio-based products are non-toxic, biodegradable, they are environment savvy, and they are good. They are not harming any environmental as well as the health. So that's why this is a particular thing which are actually required for successful bioprocessing. So the question is that yes, how this particular need can be taken care of, like is there any mean, because it is also an industrial framework. So as the awareness of the bioproducts has been increased, so their market demands is also high, so there are lots of industries which are working simultaneously and at a big level, and it is contributing significantly in our Indian as well as the global economic scenario. So so because it is an industrial framework, so one important term comes, that is the automation. Like yes, industry 1.0. So if we, because it is bio industry, we can say. So commercial bioprocessing is nothing but that is the bio industry. So if we are looking, that is the history towards the industrial framework. So industry 1.0, mechanical production equipment. So when the manual is actually taken by that is the, so when the hand is actually taken by that is the machine, so that development was that is the industrial revolution, industry 1.0. Then it is connected with that is the electricity and mass production assembly. So electricity came, then that is the industry 2.0. Then computers as well as the automated, so everything can be connected, then it is actually industry 3.0. And nowadays that is the industry 4.0 or is there, that is the cyber physical system, where the like computational digitalization is also there, but yes, everything the surrounding environment is also connected. So this is a industry how actually other industries actually grow in this manner. So based upon the requirement, based upon the need, and based upon that is the continuous requirement of the bio-based product. So it is thought that yes, This automation or this particular so industry 4.0 is actually nothing but that is the usually that is your automation trends or automation basically. So it comes towards that is the uh, it works in the principle of actually automations. So what are the driving factors in the automation in biomanufacturing? Means how what we can think like why we go for the automation. So sometimes like there is a lot of controversy that yes, if the all machines are there, so they are taking the jobs of humans. So this is a one another kind of debate which is actually going on. But simultaneously, if we look towards the benefit, there are like newer kind of jobs scenario is there, newer kind of employment opportunities for the people are there, and apart from that, how the process efficiency can be enhanced by the automation. So this particular aspect is I'm going to discuss. See, so to operate bioprocess of any degree of complexity that can be possible through automation, to improve reliability, to guarantee the quality of the product, to better deal with the process failures, increase in the human as well as the material safety, and to to improve that is the economics, means that is the cost effective way, because if the hit and trial and if like there is a more precise approach is there, so it can be suddenly that the result is actually good. So these are the like certain driving factors which go for that is the automation trends in the bioprocess. So so every like technique has two phases. Uh, some advantages are also there, some challenges are also there. So reduce human intervention means batch to batch variability is reduced risk of lot of batch, improve product quality, any kind of that is the variability is there, and increase production. So these are the major advantages of the automation processes, and these are certain challenges which has to face in the automation too, like regulatory challenges as because this has been not very new, means 5 years or 7 years old technique. So regulatory challenges is there, like certain policy, IT as well as the data integrate, because it all depends upon that is the digitalization framework, then the compatibility as well as the infrastructure, and the maintenance of that is the operational framework. So these are certain challenges which we have to face, because everything is not like every thing in the technique is not rosy always. So this is the challenges to so my particularly work focus on, because automation tools, like yes, what are the different type of automation tools which we can imply in the bioprocessing, and how the processing can be improved, how the product quality, how the process parameters, how the controlling characteristics can be improved. So because in the background we have seen all the basic aspects of the bioprocessing. So now you know that yes, at every level you need, at every level you have a checkpoint, and if is there any technique which covers all the checkpoints, so definitely the productivity or the outcome is actually quite desirable. So quality by design is a one kind of approach. Second approach is, or second tool is that is the process analytical tools, then the blockchain, machine learning, as well as that is the digital. Yes, my voice is audible.
Yes, ma'am, you're audible.
Okay. So we are talking about the automation tools. So there are like five basic automation tools which are actually implementing in the bioprocessing arena. And frankly speaking, these are not exclusively meant for the bioprocessing. These are actually used in the other industrial manufacturing. But yes, based upon their utility as well as the advancement, the bioprocessing industry has also like adopted this technology to a certain extent to enhance all the parameters which we have discussed in the last slides. Okay, so first we discuss about that is the quality by design approach. So as the name indicates, quality by design means We have to get that is the quality product by designing something, means as the name indicate. Now just try to understand this is the conventional product development and supply system, and this is the QBD approach, means quality by design approach. So if you see, that is the product development, fixed batch manufacturing process, fixed packaging process, product quarantine, and product distribution. So this is like, that is the conventional way, that how the when the product is developed, how the things are actually going. Patient means that is the end user. So as a medical fraternity, so that is the patient, but yes, you any kind of, that is the end user, any kind of like, that is the customer, you can think of. Quality system. So you see, quality sector, or you can say that is the quality control, is one of the important parameters in the bioprocess, because everything, if your raw materials are not good, your process equipments are not good, if your parameter, if your like surrounding environment is not good, it all affects actually overall quality of the product. So definitely product quality is actually getting compromised. So quality by design approach. So see, if you see the difference, product development, responsive batch or the continuous manufacturing. Responsive batch means each and every, if any defects in, that is the manufacturing processes. Responsive means you can either rectify immediately, or you can like, you have an idea, yes, if the process is going in a wrong direction, means that is the inline as well as the online data is generated through the advanced sensing technologies or the like certain systems. So you can get, that is the day to day, or the like hour to hour, or the day to day data, anytime, anywhere. So so the actual response can be actually generated. Responsive packaging process, again, all things are not key. When the system has been, when the process has been done, then you can audit and then find out the mistakes and then rectify. So this will take like huge time, also infrastructure, also you have wasted. So there is no, so if if you want to improve, then your scope is to like, that is the next batch. Yes, in this batch I have added something wrong, and then this process is wrong. So from the next batch we will improve. But here, that is the quality by design approach. There is something different. You have a responsive, means if it is a probability of improving in between, so you have, and that is the option. And this is done through the different like combinations of different tools as well as the technology, advanced technology. So it is not happens in a single day. So responsive packaging process, then the product distribution, this is like, that is the end user. So this is a particular design space, control strategy using process analytical tool, and real-time release testing. Means all things are actually gone, and that is the real-time monitoring. So basically, if you take that is the crux of this particular slide. So if you say that is the, yes, quality by design means your product, you can design the product based upon, that is the higher standard, higher quality, by implementing certain tools as well as the techniques. So what is that is the, and what are the parameters which you need to monitor, or real-time monitoring can take place? There are like, that is the certain terminologies which are actually associated. It is QTPP, is actually known as quality target product profile. Means what are your quality target, means what you want in your, that is the product, means which type of product which you are want, like which certain particular, means quality target product profile. Then CQ is known as critical quality attributes. CPP is critical process parameters. Then the design space means like you are creating by checking all these CPPs, CQAs as well as the QTPPs, you are creating a certain design space, so the whatever the probability of any kind of mismatch or any kind of deviations can take place, it can be actually rectified through the different control actually strategies, and these are done by actually, that is the process analytical technology. So as I indicated earlier, there are 5 kind of actually automated tools. So quality by design as well as the process analytical tools are actually integrated kind of approach, means they are working actually synergistically. Okay. So if you see, that is the life cycle of the quality by design. So it starts with, that is the process development, and it ends with, that is your product characterization. So again, in a very simplified words, so if the each and every checkpoint related to the from the process development, so you have seen in the previous slide whatever the different bottlenecks or parameters that need to be the bioprocess development, and then what we need as upstream as well as the downstream. So basically what happens, in a simpler words, QBD stands or QBD points have checked all the upstreaming part as well as, that is the downstreaming aspect. So 8 to 10 steps of the unit operations are from the upstreaming, and 8 to 10 steps are in, that is the downstreaming. So it covers, that is the, so it's a kind of, that is the holistic approach, or it's a kind of complete approach, or it is a kind of, that is the knowledge based management, as well as the assessment of, that is the, whatever the risk, whatever the anomaly can take place. So the product can, your desired metabolite or desired product can be your quality product, or that is the meeting the standards of actually. So in the particular, in the medical field, the medicines or the uh, like you people very much focused upon, that is the quality. So actually this quality by design term actually has been introduced from pharma. So bioprocessing industry has taken this particular term from, that is the pharmaceutical industry. So quality by design approach is there. Then process analytical technology. PAT is known as process analytical technology. As the name indicates, process analytical technology means whatever the process which are going on inside the reactor, what you can see means there are different purview of seeing the process. Online means, yes, certain parameters you need to visualize, that is the online. Certain inline means certain sensors or certain probes are actually immersed inside the reactor, and you are seeing the parameters. At line means you are not connected with online versus inline. Yes, that is the at line monitoring of particular parameters are there. Okay, you can easily visualize, and then certain analysis can be done offline. Yes, you can take the sample, go into the lab, and analysis can be takes place. So if you see this particular diagram, and I am saying that yes, for the online there are different types of tools or sensors are there. There are like that inline. There are different types of tools and techniques are there. At-line sensors are there, offline sensors. So these are actually known as, that is the process analytical tool. So if the like four-dimensional view of the process analysis can be done, so definitely the outcome of the product will be or in a desired manner. So what in the simplified words, that is the that is actually known as, so technology which deals with, that is the rigorous analysis of parameters through the advanced statistical computational algorithms, as well as the advanced imaging, as well as the spectroscopy based tools. So this particular technology is actually known as, that is the PAT. And if you see, they get different types of, you can see real time as well as the inline tools are there, as well as this is your all multivariate statistical analysis tools, like principal component analysis. So I already stated in my previous slide that yes, these are nothing new for particularly bioprocessing. These are already well established in other industrial manufacturing. So when the need is uh, considered by the bioprocess industry, so that they need to follow, yes, can we adopt such a certain advanced automation technologies in it, in bioprocessing or the biology area too. So these are just, that is the certain implementation, because the one important drawback of the bio-based industry is that they are not ready to actually change, because living cells are quite complex. So whatever they have standardized, so they want to go or they want to proceed in that way only. So it is very actually difficult to like adopt, and because it has to collaborate the data, it has to share the data, and biologist doesn't want to share the data in a very easy manner. So mindset is different. So these are actually process analytical tools which is actually used. So one example actually I have come up with, if you different biopharmaceutical means high-end pharmaceutical products like vaccines actually designs are there. So how different type of biopharm PAT can be implemented. So if you see, that is the development of PAT monitoring of CPPs like critical process parameter, digitalization, then different types of various implementing the PAT into various manufacturing processes through different tools, and then analysis can be done, different modeling can be done, so the entire process can be uh, easily optimized. So similar line, so you can say that is the near infrared scopies, mid infrared NMR, that is the Raman spectroscopy, differential light spreading. So you can check the different product quality with these and find out, yes, that yes, for my purpose or for desired purpose, what product is actually suits you. So this is the process analytical technology particularly. Then comes that is the blockchain. So to be very frank, blockchain is actually, it's a actually financial term. So it is a banking term which is used, and you are very well versed now with the cryptocurrency, bitcoins and all. So the question is, or the surprising thing is, that how blockchain and blockchain technology is actually used in biomanufacturing arena or bioprocessing. So if we can, like biology, we can be benefited with the blockchain. So yes, this is a like major wonder of the blockchain. So first we need to understand that blockchain is nothing but like I must say that it is a kind, though it is written inside the slide, it is a kind of actually information which is stored in many computers, but in a secured, and nobody can actually alter. So once you have data feed inside one system, then it will be like displayed same data in the multiple computers or like thousands of computers, and without alteration. Means if you want to alter, if you want to like erase the data, if you want to modify the data, if you want to delete the data, there is no scope. So it is a kind of peer-to-peer connected, or you can say that is the end-to-end encrypted data storage, or the but because it is a financial term, so if you see my this particular slide as the introductory slide, so you can see that yes, one transaction is requested, so the transaction is broadcasted to peer-to-peer network. The network is like, this is actually blocks, different blocks are there, so information is actually compiled in different blocks. So a digital database or ledger that is distributed among the nodes of a peer-to-peer network. So whatever the and networkers are like so many, well, thousand of networks are there. It doesn't be like 3 to 5 people. The information is secured between 3 to the information is secured, and as such precise, and whatever so if your data, if your entry is correct, then it will be displayed same entry whether you are sitting in the India or sitting in that is the abroad. So it is like such kind of technology is there, and without any consent, means without nobody can alter that particular kind of information. So it is a kind of secure actually networking network, and it is a transparent system, that yes, once the information has been like information is released, so there is no chance of like erasing or uh, like uh, uh, like altering any kind of scope for that is erasing the information. So verified transaction, and then the transaction is combined with either transactions. Once verified, new block is added, and then the transaction is finished. So this is like one kind of framework. So I already told now, this is a banking or the financial term. So how this particular blockchain process is actually connected for our purpose. Again, it is a more comprehensive, like one it is buyer and one is seller. So for our, as we are also industry based, a bio-based industry, so some customers are there and we are seller of the bio product. So how our transaction, so one term is like that is the mediator. So there is no scope of actually mediator. There is no scope of any creating misinformation and making any kind of like that is the money inside by the mediator. So this particular buyer is there. So whatever the transaction is actually given. So it is secured in is in a digital database. Then the crypto hashing is there. Then it is distributed. To transaction is committed to blockchain and miners are rewarded and seller receive, that is the transaction. So all channels are actually transparent. Everybody knows that yes, what the buyer as well as the seller ke beech ki whatever the dealing is there, it is actually transparent at every level. So this is an idea behind that is the blockchain. So these are the different actors, or means like these are like certain driving factors, or like whatever the major stakeholders in the blockchain technology. So see, blockchain user is one is, and blockchain network operator means because it is a completely digital framework. So definitely high-end computing facility is there. Blockchain developer is needed, architect is needed, regulator is needed, then the processing platform, then the data sources, membership, as well as that is the final, that is the user. So these are actually what we actually required in that is the blockchain. Now this is for particularly, I keep this particular information, how the blockchain actually came into the limelight. So during the COVID period, for the medical fraternity, we know that yes, how much COVID patients are actually infected with that in the surroundings. So there are lots of apps. So how this information can be actually communicated? So this is actually all due to your blockchain technology. So infection verified subsystem, here that is the confirmed COVID-19 cases that is stored in that is the blockchain data, and it is like communicated with different blocks, means like in the how the India knows that yes, government knows that yes, in the UP, daily COVID cases are there, in the MP, that is. So all information is actually peer-to-peer, end-to-end encrypted and precise, and that is well connected, secured. And if the if that particular information is there, the data has been tracked, mass surveillance system is operated, and that those with the confirmed cases, they are actually quarantined, they are actually informed, or they are taking proper care of by that is the at that time. So you have all gone through, we have all gone actually, we all gone through this particular phase. So when I gone through that, yes, this why how this particular system works. So the success behind that is the blockchain. So it is that blockchain technology plays very important role. And for the medical fraternity, counterfeiting of the drugs are also one of the major challenges that doctors face or the medical fraternity face. So counterfeiting of the drugs can also be effectively solved by that is the blockchain. Now my purpose is here to how the biomanufacturing data, means bioprocessing area, how the blockchain can be implemented. So this is a one kind of mind map where the 5 segments are there, like different technologies of the blockchain, application, maturity of approach, reason for using blockchain, as well as that is the biomanufacturing data. So let's talk about that is the different technologies of the blockchain. So if you see, Bitcoin and Ethereum is like most popular, but yes, this is also multi-chain, Tendermint, Juice, Gcoin, NAM, ITA, Custom. These are all the different crypto, like this is all that is the digital platforms for doing that is the blockchain. So but yes, Bitcoin and Ethereum is like more popular, but yes, these are the different systems. Then how biomanufacturing data, so as we are aware of this, that is the upstream process as well as the downstream. So any kind of lots about like peak data, storage, packaging, transportation. So these are all can be making different blocks, and that can be well connected to each other. So the proper information can be actually traced down. Upstream, similarly in case of that is the upstream, raw material collection, shake flask, whatever the readings or whatever the conditions for the shake flask is there, what are the total time, temperature control, media optimization, how the reactor function in a proper way or not. So for a particular process, so these are all that data can be stored in a secured as well as the end-to-end encrypted manner. Then reasons for using blockchain, that is the disintermediation, data governance and management of records, internal process management too, and that is the provenance also. Then it is a mature kind of approach. So it has like that is the architecture, implementation is also possible. Evaluation, we can do evaluation. We can make like that is the proposal for that is the advancement. And then what are the applications? So handling research and development data for our biomanufacturing, transaction records for, yes, because it's an industrial framework, product, our bioproduct safety, our cyber security, whatever the data we have, regulatory compliances are there, as well as that is the proof of concept. So this particular mind maps covers that, yes, how different blockchain technologies can be helpful in the biomanufacturing or the bioprocessing actually area. So that can be like these areas can be like very much rigorously complied with blockchain technologies. Now, one example I have created, like manufacturer in regional area as well as major manufacturer in that is the country. So that is within the country also, as well as that is the outside the country also. Like means like that is the local manufacturer and that is your the large scale like commercial manufacturer are there. So how this upstream versus that is the downstream data can be, so you can see these are like different, these are acting different actually blocks, and every information is stored in different blocks, and then this will be like verified by the different kinds of regulatory compliances and the verifiers, and then you can like your information is actually transparent each and every, if like where you bought from that is the raw material, what kind of quality of your raw material, so that is information is stored in one block. So verifiers can verify, and that information can directly supply to that is the major manufacturer. So such a kind of transparent culturing conditions, like what you have chosen the culture conditions for your process, like whatever shake flask parameters, how the bioreactor scale up has been done for a particular product, and then how you harvest, and then the further purification, formulation, storage condition, whether you have performed, what quality check you have performed at what date. So everything is stored, and what are the transportation, like where your product is, when it is delivered, when it is coming to the market. So these are all like interconnect. So you can see, so this particular like that is the interconnection of network. So the actual and it is also the information is also retrieved by that is the regional area. So your like job is very much easy. You don't have to like that is the hassle that yes, where my product is there, where it is reached in a proper place or not. So by making the digitalization or by making the such kind of that is the automation workflow, you can have such kind of like wonderful information for in the area of that is the bioprocessing. So one example I have taken for that is the biorefinery. So as the oil refinery is there, so one normal term or one common term is on the biorefinery. Biorefinery means apart from your biofuels like bioethanol, bioetanol, various kind of platform or the specialty chemicals can also be retrieved when the fermentation is carried out. That is the different type of lignocellulosic biomass. So it doesn't only key, yes, you can process this particular biomass pre-treatment fermentation, and then you can only make that is your biofuel. No, there are lots of scope from the waste, that variety of the biorefinery means multiple products at a time, means refining of multiple products at a biological scale or from bioindustry. That is actually known as biorefinery. So here how that is the blockchain is working. So if you see that the verifier has all knowledge, yes, where you which type of raw materials are there, what is the raw material actually you have taken, then what are the pre-treatment methodologies are there, like whether you treated with the alkali to ferment enzyme, then acid, then you put the system into the reactor, which kind of microbes, then your product recovery, how it is done, what is the transportation is there, then when where it goes, that is the utilities, where that is the waste management is there, and ultimately all these things are actually automated and the information is secured. It is end-to-end, or that is the peer-to-peer networking. So not only by verifier, you can like put any kind of other stakeholders too, like you can draw, you can connect this information to other stakeholders too. So this information is same for this, you cannot erase. So once the information is generated, once the information is there, you put inside the computers, it can be stored as such. It is neither read nor erased, nor so it is like that is the secure data inside. So this is a kind of actually blockchain framework. So still there is a like we can say it is not very matured on till date, but yes, certain applications have been seen. In the medical science, there are like counterfeiting of drugs, vaccine distribution. The blockchain plays very important role. Mass surveillance system, I shown in one slide. It is very, but in the bio manufacturing area, it has been actually gaining momentum or gaining like it is comes into lots of still blockchain is yet come to play that is the in a major role. So certain benefits are there for the blockchain manufacturing as well as certain threats, because see every technology has certain 2 points of technology is there. So certain benefits also and certain that is the threats also, because it's a newer concept. So technical skill, that is the scalability issue, high energy consumption, social acceptance, because people don't know, people are very much frightened about that is the data sharing. And sometimes they are not actually mentally they are not ready to like share everything to the supplier, everything to the buyer. So so there are lots of issues for the social acceptance as well. But yes, the technology is quite promising, and I think in future in the bioprocessing, the blockchain can be a good for making that is the digitalization data and making the process more transparent and secure. Okay, next aspect or next automation tool is that is your machine learning. So today that is the current buzzword is your AI, artificial intelligence and machine learning approach. So artificial intelligence is a broader term. Machine learning is a kind of actually artificial intelligence platform where you have certain, you input certain data, means you train the machine with a certain input data, whatever the generated or whatever you have, and then machine automatically with the over of time, machine automatically learns to to infer, like find patterns. So see what is the how machine learning works, like input data, analyze, find patterns, means what are the current trends. Suppose there is a temperature range today, that is the it is predicted that yes 45 degrees. So the last 10 years the same 19th of July, that what is the, so it creates one kind of actually pattern. Then certain predictions are there, and then that is there, feedback is there. Yes, in the next coming years such kind of possibility, such temperature range possibility is there. So this is a machine learning. So how actually machine learning actually works for now. So in the if we are talking about like there are certain, because machine learning is nothing but that is the advanced computational algorithm to sort out the data, to analyze or to channelize the data, and to come with certain actually outcome. Okay, so there are three types of actually machine learning protocols are there: supervised, unsupervised, as well as that is the reinforcement. So and among the supervised, there is variables, continuous, categorical, variable not available. Unsupervised means like there are no variables are present, whatever the random data is there, and you can use for that is the unsupervised. Reinforcement, like optimization as well as the control, and these are all MLR and PLS, KNN. These are all actually different kinds of actually method or the methodologies of that is the different machine learning process. So in the bioprocess, so I'm not going to detail with these processes because it is uh, it has a separate like description. But how this like the concept is to show that how the automation or the machine learning is involved in the actually bioprocess. So see, if you can see strain selection, the ML approach can be used. Then the bioprocess optimization we can use the machine learning. Then the bioprocess scale up, the machine learning data approach can be used, as well as the controlling of the bioprocess, whatever the parameters can be altered during the process, how they are actually controlled, how they can be monitored. So these are like that is the particularly aspects in the bioprocessing where the machine different like algorithms of the sometimes supervised, sometimes unsupervised learning approaches can be actually used, and by combination of these, like how the complexity of actually biological system can be actually reduced. Okay, so this is like that is the core concepts how this particular machine learning framework can be used, and I have come up with certain examples like role of machine learning in biorefinery. Means again biorefinery, I told you that one kind of like factory or the refinery where the multiple biological products can be retrieved from different biomass. Okay, so different biomass like agricultural residues, forest waste, different kind of grass, or any that is the industrial waste. So machine learning, how it is helpful. So whatever their composition is there, so the data is so their pattern is generated like biomass feed platform chemicals. These are actually platform chemicals, and these are actually value added chemicals which can be formed. So machine learning, what we matlab, how this machine learning can be implemented ki when this particular data ki yes, this particular agricultural residues can produce the levulinic acid also at certain conditions, sorbitol also at certain condition, itaponic at certain conditions, sulfuryl, and that lead to the production of like biofuels also, biomaterials also. So when the voluminous, because biorefinery is also so large amount of data is generated, so when it is feeded inside the machine that yes, such kind of residues have such kind of pattern, so automatically it predicts it gives an idea that yes, if the such kind of residues are there, such kind of probability of getting product is certain amount of getting this particular product is more. So this in this way ML is actually helpful in biorefinery process. Then role of machine learning in that is the development of monoclonal antibodies. So see, lots of work has been actually done in the computational framework. Nowadays what happens, all works are actually done in that is the in silico way. Why? Because to reduce the time, to reduce the to save the cost, to preserve our infrastructure, to preserve the energy. So what happens? So if the certain kind of if you see, yes, monoclonal antibodies, cetuximab. So these are like your monoclonal antibody. These are actually different kind of actually marker antibody specific properties, like what is the binding affinity, what are that is the marker, what are EGFR, or what is their effectiveness. Then these are actually finalized parameters कि भाई क्या होगा, molecular weight, glycosylation pattern, what is the protein content in the antibody, then what are that is the light chain composition, what is heavy chain composition, what is the cytotoxicity, as well as that is the heterogeneity. And then based upon different, these are nothing but that is the network means these are certain algorithms of the ML or machine learning that helps the, yes, if this particular antibody have to take, the binding affinity is such, and that binding affinity is actually determined or is affecting that is the protein content, or that is the glycosylation pattern, or that is the molecular weight. So you can estimate, so yes, that what kind of molecule which can be retrieved, or what kind of designing of your monoclonal antibody can be done. So it gives an idea or predict that yes, such kind of novel molecule can be generated, and definitely you have to validate in the wet lab, because all things cannot be done through the computational studies. So in the biology there is always scope of actually wet lab studies, means you have to validate inside the lab, but yes, lots of hit and trial approach can be actually saved. So it is very effective nowadays, and people are working immensely on that. Okay. Then role of ML in combating AMR. So as we know that we are suffering from that is the like major global threat, that is the antimicrobial resistance, and you know very well this phenomena. So how this if we are like looking for a development of your antibiotic, Okay, so it took years because to find out the novel antibiotic, to find out like novel antimicrobial compound in a same efficacy, it will be a difficult task. So what is there, like there are various types of actually public databases, drug banks are there, PubChem molecules which are zinc databases. So these are like databases of the chemical compounds. Then they're all physicochemical properties are there. Then the machine learning model we can apply, and then we predict that yes, more fast. Like there are already ADME properties, there are certain tools are there already existing, conventional tools or the computational tools. But by machine learning, you can have a more comprehensive knowledge like drug likeness prediction.
Excuse me, ma'am.
Prediction as well as the novel molecule. Yes.
I'm sorry to interrupt.
Yes.
Ma'am, how long you'll be taking, ma'am?
Just to 5 to 7 minutes, 10 minutes, ma'am.
Okay, ma'am. Yes,
Yeah.
yes.
Well, my last digital twin is remaining automation tool.
Sure, ma'am. Yes.
So this role of machine learning in that is the protein engineering. So if we go for like how the novel enzyme can be created and in an in silico manner, means different enzymes which are already structure information is there. So by combining the different machine learning models, tools and algorithms, different predictions and analysis can be done with the enzyme, like catalytic activity prediction, enzyme key stability, substrate specificity, protein insolubility, and then the commercial production. Means like lots of actually information which is actually not covered by your traditional computational tools, that is actually more fast and more rigorous, more appropriate, is actually done by that is your machine learning process. Then the last automation tool is actually digital twin. So as the name indicates, that is the twin, means it is an interface between the real world versus that is the virtual world. So it is a kind of actually virtual system which is actually giving a real information from the real world or from the real space. Okay. So a digital twin is a comprehensive integrated mathematical model based on historical and current data that stimulate a complete bioprocess sequence in real time. So you can better understand that this particular diagram. So see, plant model, this is a complete factory or the biomanufacturing plant. This is your process which are going on. This is the product, and this is like that is the like end use customer or that is the patient. So building layout, building modules, equipment and utility models, module properties, how the infrastructure is there, HVAC, high ventilation, air conditioning, material energy flow. So this is the different plant model, whether it is plant fit for the process, see process fit for the product and product fit for the patient. So this is complete, it is a kind of like that is the holistic involvement of all four. So till whatever we discuss, we cannot cover that is the infrastructure facility, but digital twin actually, if you see, it also cover that is the space where you actually are working. Till date, we are focused on that is the only the process as well as the product, whether it is in the quality by design or whether it is by that is the process analytical tool. So, but here we by implementing the digital twin, you can focus on that. Yes, whether the whether your like a utility system, whether your infrastructure is good for the process, whether your process is fit for the product development, and whether your generated product by so these are the all product qualities, these are all patient or the customer information, and that information is actually all things are taken care of by that is the intermingling between the virtual as well as the real world. So if you see this bioprocess digital twin platform, this is your virtual, this is your that is the real, means virtual system, all data is generated through the virtual or the simulations if you have heard, and this is your real time inline data monitoring. So one reactor is at one time that is the simulation is going on and the data is actually interconnected. So if you see that the data is actually interconnected, so if any kind of like anomaly is there, any kind of disturbances, any kind of variability occurs, so you immediately actually have to rectify, means you have a scope for actually immediate correction for that. And anytime, anywhere, you can retrieve the online data, or that is the inline data. So there are the advanced, very advanced platform for bioprocessing. So this is the just yes, from inoculation to formulation of the product, how this digital twin can be working, plant simulation, plant analysis, as well as the plant optimization. So this is all about, that is the automation in bioprocess. And this is the just recently published paper in Nature Biotechnology. So they said that yes, one of the future of the bioprocessing is, that is the space bioprocess engineering, where so space is no new to the bioprocess engineer or the doctor's fraternity. So to resource utilization, to find out the different food as well as the pharmaceuticals, in situ manufacturing, as well as the system design. So I liked it to share that yes, space bioengineering is one multidisciplinary approach, or it is a like biologically driven process technology, where the different modalities have been actually found in these space too. So not in the Earth, space has also scope of actually bioprocessing now. So this is one. And end-to-end connected means where the biopharma 4.0 convergence between the processing automation as well as the software technologies, means that is the smart labs are there, where end-to-end connected bioprocessing is there. So this is the future aspect. So maybe I think very soon we will see the such kind of industrial framework in the bioprocessing. Yeah. So this completes my talk, and thank you. So the session is open for questions. Is any question?
I request the participants to post questions in the chat box, or you can raise your hand. No questions, sir. No questions. Ma'am, there are no questions as such. If any, will mail you or text you, ma'am. And it was a great session. Thank you, ma'am, for such an informative session on automation in bioprocessing. Thank you, ma'am. Now I would like to propose vote of thanks. Good afternoon, one and all. I take this opportunity to propose vote of thanks on behalf of Department of Biochemistry, Shri Devaraj Medical College. First and foremost, we salute our late founder, President Shri R.L. Jalappaji, who continues to be a beacon of light. We would like to thank our patrons of Shri Devaraj Education Trust, respected President and Honorable Chancellor Shri G.H. Nagaraja, respected Honorable Vice President Shri Rajendra Jay, respected Secretary Shri Hanumantaraju KG, in charge Honorable Vice Chancellor and respected Registrar Dr. DVLN Prasad, Dean Faculty of Medicine and our beloved Principal Dr. Prabhakar K, respected Medical Superintendent Dr. Krishnappa J, and I thank Dr. Shashidhar K. Sir, Professor, Head Department of Biochemistry, SDMC, for his continuous support and encouragement. I also extend my thanks to other officers of the Academy. I thank all the faculty members and stakeholders of SDHR for their enthusiastic participation in the event. Last but not least, I thank the non-teaching and technical team members who are part of this program. Thank you one and all. Thank you so much, ma'am, for accepting our request and giving us such a great session. Thank you.
Thank you so much. Thank you, Dr. Soumya.
Yes, ma'am. Yes.
Thank you all for wonderful listening. Yeah.
Yes, ma'am. Shall we end the session, ma'am?
Yes, yes, yes.
Sir.
Thank you.
Thank you, all the participants. Can you please switch on the camera so that we'll take a pic? Oh, madam. Thank you, one and all. Leave.